A very good afternoon to all friends. I am Dr. Varunendra Singh Rawat from the Department of Zoology, Hindu College, and today I will be delivering a lecture on DNA damage and repair. In my previous lecture, I talked about the methods by which DNA is damaged, which leads to a change in the information coded by DNA, and this leads to faulty replication or faulty transcription and subsequently faulty translation. So I talked about all the methods by which DNA is damaged. Today I will be focusing on methods which lead to repair of such damaged DNA. Because in any cell it is essential that whatever damage has occurred to the DNA which contains all the information, genetic information, it should be repaired before that is passed on to the subsequent generation. So my focus today will be on the repair mechanism, but I will start with a brief summary of the various mechanisms which damage DNA. Now all these mechanisms which damage DNA are collectively termed as mutations. These mutations could be germinal or these could be somatic mutations. Somatic mutations in eukaryotic organisms occur in the cell of the organisms, normal body cells of the organism, whereas germinal mutations occur in the germ cells. Only the mutations which occur in germ cells are passed on from one organism to another organism in sexually reproducing organisms. Whereas in unicellular organism or prokaryotes, whatever mutations occur in the DNA of the organism, they will pass on to the subsequent generation. There is a mutation rate which occur in different types of organisms, which depends on all the different processes involved in replication of DNA and repair of DNA. If there is no mutation, then there will not be any evolution of new species. And if the mutation rate is very high, then it will be very detrimental to the organism and the subsequent generations as well. And that particular species which accumulates very high number of mutations might get extinct in a short duration of time. So mutations can be categorized. They could be arising due to incorporation of wrong nucleotide, due to damage to DNA, which could be spontaneous or induced, or due to illegitimate recombination, or due to insertion of mobile genetic elements, which are also known as jumping genes. And they could lead to damage to single bases or misincorporation of bases, damage to a long stretch of nucleotides, or damage to the double-stranded DNA, and this could lead to a very high mutation rate. The fidelity of replication is ensured by DNA polymerase because it has many such domains which ensures that there is no mismatch during replication, but still mutation do occur and there is misincorporation of bases during replication. Now the spontaneous chemical changes in DNA occur because of the chemical structure of the bases, and this occurs because of the shifting in the hydrogen atom in the bases, and this leads to formation of tautomers. These tautomeric shifts lead to formation of tautomeric bases which do not have the base pairing ability like the normal bases. Like there could be keto form or enol form for thymine and keto form and enol form for guanine, in which case the keto form are the normal form and enol forms are the aberrant forms or the tautomeric form. If their tautomeric forms are present, then they will not be base pairing with the normal bases. For example, if guanine is present in keto form, it will be base pairing with cytosine, whereas if it is present in enol form, it will not base pair with cytosine. As can be seen in this particular figure, there is altered base pairing of different bases whenever there is tautomeric shift or the bases are occurring in tautomeric forms, and they do not obey the Chargaff's rule that A should pair with T and G should pair with C. As can be seen here, after one round of replication, this mutation or this mismatch incorporation will be fixed in the particular cell or the organism. Like as can be seen in the upper hand of the upper part of the diagram, G is base paired with T, whereas it should have been base paired with C. Now whenever this particular DNA replicates, there will be change from GC base pairing to AT base pairing, and that will be a mutant organism or mutant type of cell. And if this particular mutation leads to change in the function of the gene or the protein that could be detrimental to the organism. Now, there are certain mutations known as frameshift mutations which occur whenever there is a repeat of dinucleotides or trinucleotides in the sequence of the nucleotide, in the sequence of the DNA. This di or trinucleotides, when repeated a number of times, leads to slippage of the replication machinery. So much so that this dinucleotide or trinucleotide region might be increased in number or it might decrease in number. And many human diseases, genetic diseases, are caused because of increase or decrease in the number of these di or trinucleotides. For example, Huntington disease, muscular dystrophy are some of the diseases which are caused whenever there is increase in the number of CGG or CAG repeats in the DNA of the organism. As can be seen in this particular figure, there is slipping of the replication machinery which leads to incorporation of additional bases, and this in turn will lead to change in the coding sequence of the gene, which will lead to change in the sequence of the mRNA, and this will subsequently lead to change in the amino acid sequence of the protein. Whatever damage occurs to DNA is known as DNA lesion. Now, DNA lesion could be misinstructional lesion or non-instructional lesion. Misinstructional lesion occurs whenever a wrong base gets incorporated in the DNA, which codes for a wrong base in the transcript, which subsequently leads to wrong amino acid getting incorporated. Or there could be non-instructional lesion wherein there is a block, replication block, so that the machinery for replication cannot proceed on further, and there is no passing of information from one generation to next generation. Now, these lesions could be arising because of the intrinsic properties of the DNA, or these could be arising because of the radicals which are formed during various metabolic reactions in the body, and these radicals harm the DNA, changing the consistency of the bases, and this could also arise due to replication and recombination errors, and also because of insertion of the genetic elements also known as transposon. Induced lesion occur whenever the organism is exposed to agents which are mutagenic or ionizing radiations. These mutagenic chemicals or ionizing radiations cause damage to DNA, which leads to changes in the structure of the bases and leads to mutation. The various categories of DNA damages are incorporation of illegitimate bases, that is, bases which are other than A, G, C and T bases present in DNA of the organism. These bases could arise because of spontaneous deamination or because of any physical or chemical agents or incorporation of base analogues. Base analogues are chemicals which are similar in structure to bases, but they have different pairing capability than the normal bases. Also, whenever there is addition of side groups, bulky side groups like methyl groups or ethyl groups, this changes the chemical consistency of the bases, and they do not pair with the normal base pairing method. Like in this particular example, we see that cytosine, when deaminated, it leads to formation of uracil. Uracil is not a natural base in DNA. Uracil is found in RNA. The presence of uracil in DNA suggests that there has been some damage to the DNA. Likewise, whenever there is deamination of adenine or guanine, it leads to formation of some aberrant bases which have different pairing ability. As can be seen in this particular figure, there is C cytosine pairs with guanine, but whenever there is deamination, it leads to formation of U, which is uracil, and uracil will lead to incorporation of A in the next strand, which is getting synthesized. So there will be change from GC coding to TA coding, and so it will happen with all the other bases which are getting deaminated. Oxidation of bases also causes changes in the structure of the bases. Like in this particular example, there could be oxidation at the 8th position, or in like 7,8 dihydro 8 oxoguanine, there is oxidation at 7 and 8 position, and this lead to alteration of the pairing of guanine. It does not anymore pair with C, instead it pairs with A. So this is one of the methods by which bases can get modified. Also, alkylation could lead to addition of ethyl group or methyl group, which again have pairing capability different than the original bases. Like in this particular example, it is seen that addition of alkyl group changes the pairing of guanine, which normally pairs with cytosine, now it is pairing with thymine. So, this is one of the methods by which bases get modified. And there are base analogues. The base analogues are those chemicals which have structure similar to normal bases, but they do not, they might get incorporated in DNA, but they do not code for the normal complementary base pair. A will not be pairing with T, it will be pairing with some other base. In this particular example, bromouracil, which is in keto form, it pairs with adenine. It's a base analog of thymine, but whenever it is present in enol form, it doesn't pair with adenine. Instead, it pairs with guanine. There are intercalating agents, which are large bulky molecules which get intercalated between two base pairs, and they can lead to addition or deletion of the bases whenever DNA gets replicated. Ethidium bromide, proflavin and acridine orange are example of such bases. Then there can be formation of pyrimidine dimers whenever organisms might get exposed to UV light. There can be thymine dimer formation or thymine cytosine dimer formation, and these particular dimers lead to formation of a replication block, because DNA polymerase cannot pass through these particular dimers. Other type of DNA damages include formation of apurinic or apyrimidinic sites, wherein the base which is attached to the particular deoxyribose sugar gets removed, and this particular base does not have a purine or pyrimidine, so it cannot code for a complementary base pair, or whenever transcription is occurring, it cannot code for any nucleotide at that particular position. Other type of DNA damage is formation of gap, that is whenever there is a stretch of nucleotide missing from one of the strand of DNA, that is known as gap, and whenever such gap occurs, it could lead to block of the replication machinery or loss of information, so that during replication or transcription there is no specific information related to that particular gap. And a nick can occur whenever there is a break in one of the strand of the DNA, though there is no deletion of nucleotide as such, but the strands are not continuous, and this again can cause a blocking of the replication machinery. There can be double stranded break when there is a severe damage to the cell or the organism's DNA. It can lead to breaks in the double stranded DNA. The chromosomal DNA might break, and this is a very serious type of damage which requires immediate attention by the cell, and usually it is caused by some chemical or ionizing radiation. Then there could be cross linking between strands of DNA. There could be intra-strand cross linking or inter-strand cross linking, and again this could be because of UV induction, dimerization during UV induction, and this again leads to blocking of the replication machinery. With this background, which I had covered earlier also, I move on to the repair mechanisms which are taking place in the cell for repairing all the damage which has been done to the DNA. Now, various mechanisms have evolved over course of time in the organism which help in maintaining the sanctity of the DNA, and these mechanisms can be categorized as direct reversal, wherein the damage which has been done is directly reverted back, so that you get a normal base which is not damaged anymore, or there could be mismatch repair, which helps in repairing any mismatches which have gotten incorporated during replication of the DNA. This is a very important type of repair mechanism. Another is base excision repair mechanism. Base excision repair mechanism helps in removing all the bases which are damaged or modified, so that a normal base can get incorporated in the place of the damaged base. Whereas nucleotide excision repair does not involve removal of a single base. It involves removal of a stretch of nucleotides. After that, the gap is created, and this gap can be filled by normal DNA polymerases and can be sealed by DNA ligase machinery. Then whenever organism is exposed to severe radiations or chemicals which induce severe trauma to the cell and bring about double stranded break in the DNA, double strand break repair mechanism come into play. This help in joining double stranded DNA which have broken, so that no information is lost, and even if some information is lost, still the two ends are joined, two ends of the two strands of DNA are joined together, so that replication or transcription machinery can move on. Because if the double strand break is not repaired, the replication or transcription machinery won't move from the point of breakage, and that will be very serious condition, because there won't be any replication, there won't be any formation of RNA and subsequently proteins for that particular region of DNA. So, cell tries to join the two broken end of the DNA, even if there is some loss of information from in between. And translesion DNA synthesis is that type of repair mechanism which comes into play when all the other mechanisms have failed. It is a part of SOS response for the cell. When there is severe damage to the DNA, translesion DNA repair mechanism come into play. Now, coming back to direct reversal mechanism, the simplest of these mechanism is ligation of nick. Whenever a nick occurs in any strand of the DNA, that is, it is not continuous and it is broken, but still it has 3' OH group and a phosphate group at the 5' end. Therein, there will be repairing. Repairing will be done by ligase, DNA ligase, which will join back the 3' OH group and 5' phosphate group, so that the nick which has gotten introduced in the DNA strand, it gets repaired and there is no more break in the DNA. The second type of reversal mechanism is photoreactivation repair mechanism. Whenever organism is exposed to UV light, then as I talked about earlier, there is formation of dimers, pyrimidine dimers. These could be thymine dimers or these could be thymine and cytosine dimers. These dimers present a replication block. Replication machinery cannot move forward from this particular site. Now, in order to monomerize this particular dimer, cells have an enzyme known as photolyase. Photolyase in the presence of visible light, that is light between 300 to 500 nanometer, leads to monomerization of the dimer. So that now we are having a normal thymine base pairs instead of a thymine dimer which exists as a cyclobutane ring, and there is no more replication block present here. But this occurs in the presence of visible light only, and it is also termed as photoreactivation repair. So, it does not require any serious changes to the DNA, it only requires presence of UV light and photolyase enzyme which will be converting dimer back to the monomers. Another type of repair mechanism which comes under direct reversal mechanism is removal of alkyl groups from the bases. Alkylation is a major process which occurs in the cells of the organisms, in the DNA of the organism, and it occurs in all the bases. In this particular example, we see addition of methyl group to the guanine at oxygen 6. This methyl guanine, instead of pairing with cytosine, it pairs with thymine. Now, whenever this type of change occurs, it will lead to mutation, change of information. So, the cells have developed mechanism for removing these alkyl groups. One of the common mechanism or common enzyme which removes the alkyl groups are methyl transferase enzymes. These methyl transferase enzymes remove methyl groups from the bases and include them in the cysteine groups which are present at their C terminal end. So, methyl transferase essentially is removing a methyl group from the base and taking it up. However, during this process it undergoes conformational change, so that it is no more functional. Therefore, these methyl transferases are also known as suicidal enzymes. Whenever there is a very large quantity of methyl transferases which have been blocked by addition of the alkyl groups, this acts as a signal for the cell to enhance the transcription or the expression of the methyl transferase gene. Whenever there is severe damage, that means methyl transferase will be having more of alkyl groups, more of the methyl transferases will be inactivated, and this particular signal is for the methyl transferase gene to start expressing itself. Once there is removal of the alkyl groups, the damage has been undone, then these methyl transferase enzymes will be degraded and these degraded products will be the signal for cell to stop expressing these particular methyl transferases enzyme. A very common enzyme, methyl transferase enzyme in E. coli is ADA, which is a methyl transferase. Whenever there are alkylating agents present in the environment of E. coli and alkylation of DNA is occurring, then there is induction of some genes like ADA genes, ALK genes and ALKB gene. ALK gene is a glycosylase which breaks the glycosidic bond between the base and the sugar, and it takes part in the base excision repair mechanism. But ADA is a methyl transferase and it helps in removal of methyl from the alkylated bases, and it itself is a positive regulator of its own gene. Whenever there is serious level of alkylation in the DNA, then phosphodiester bonds also gets alkylated, and then the adaptive response is generated in E. coli, and it gets terminated. This response is terminated only when there is cleavage of the methylated inactive ADA protein. The next type of repair system which I will be talking about is mismatch repair system. The mismatch repair system helps in removing any mismatches which have gotten incorporated during replication of DNA. For this particular repair mechanism, there is a set of proteins or enzymes known as MUT proteins. MUT stands for mutator, and a group of mutator proteins, namely MUT-S, MUT-L and MUT-H are taking part together to induce this particular mismatch repair mechanism. Now, MUT-S is a protein which recognizes any mismatch which causes kink or distortion in the structure of DNA. This MUT-S protein gets loaded onto the DNA wherever there is distortion. It is an ATP utilizing an ATP binding protein. Whenever MUT-S is loaded onto the distortion, by utilizing ATP, it undergoes conformational change at this particular location, and it recruits another protein known as MUT-L. MUT-L and MUT-S form an active complex, and they in association with MUT-H induce a cut in the DNA. Now, MUT-H is another protein which is bound to GATC sites in the E. coli DNA. GATC repeat is present frequently in the DNA of the E. coli. About every 256 base pair there is a repeat of GATC nucleotides, and MUT-H binds there. Whenever MUT-H binds to active MUT-S and MUT-L complex, then MUT-H also gets activated. As can be seen in the left diagram, which is for E. coli, whenever MUT-H gets activated, it induces an incision. Because it is an endonuclease, it induces a cut in one of the strand of the DNA. After the cut has been made, there are exonucleases which come into action, and they start removing bases from that particular site. After a small chunk of this particular DNA strand has been removed, which includes the mismatch site also, then repair mechanism comes into play, and DNA polymerase starts synthesizing the DNA at that particular gap, and again there is ligation by DNA ligase. However, majority of the bacteria do not use this particular system, that is they use just MUT-S and MUT-L for inducing a cut in the DNA molecule, and after that exonucleases help in removing the bases. Now, exonucleases can proceed either from 5' to 3' direction or from 3' to 5' direction. Exonucleases which are proceeding from 5' to 3' direction, one example is exo7 or rec-J, which moves from 5' cut toward 3' direction, and in process it also removes the mismatch, and then a repair mechanism comes into play. Another example of exonuclease is exo6, which moves from 3' to 5' direction as seen in the right side of the figure, and it after removing mismatch again there is repair synthesis. So, this particular strand can be removed from both the sides. Now, how does MUT-H ensure that only the wrong strand is removed, that is only the strand which has a mismatch is removed? This is a very serious problem, but it is overcome beautifully by the MUT-H recognition system, because MUT-H recognizes hemimethylated DNA. There are GATC sites in the DNA of the E. coli, and these A in GATC are methylated. In any organism which is not undergoing replication, both the strands of the DNA are methylated at A position. But after replication, the daughter strand which has formed newly, the A residues, the adenine bases, are not methylated initially. So MUT-H recognizes these particular bases, hemimethylated bases, that is the adenine bases which are not methylated till now, and it introduces a cut only in those strands. Because the mismatches will be introduced only in the daughter strand, therefore it will cut the daughter strand only, and thus it ensures that only the correct strand is cut and and at the correct side bases are removed. Otherwise, it poses a problem because parental strand could also have gotten removed. So this is a very nice mechanism by which MUT-H ensures removal of mismatches. There are short patch mismatch repair system existing in E. coli, which are not MUT-S, MUT-L and MUT-H dependent. One is MUT-Y dependent repair, which replaces adenine residues whenever it finds AG and AC mismatches. It primarily removes by the glycosylase activity, but it can also act on mismatches. Also, there is another type of repair mechanism which is very short patch mismatch repair, which removes T residues from the GT mismatches. However, this particular system requires presence of another endonuclease which is coded by VSR gene. In eukaryotes also, there are homologs of the MUT-S and MUT-L proteins, and these are known as MUT-S alpha, MUT-L alpha, and they carry out their work by a similar mechanism. They induce a cut and DNA is removed, one of the strand of DNA is removed, and DNA polymerase delta and DNA ligase fill in the gap. It has been seen that MUT-S alpha and MUT-L alpha, whenever there is mutation in these two genes, it leads to cancer. One of the very common cancer known as hereditary non-polyposis colorectal cancer is introduced in human beings by mutation in MUT-S alpha and MUT-L alpha genes. So these proteins are very much required for mismatch repair in human beings. How do these proteins recognize that which strand is going to be cut? Because there is no such GATC sites, methylated GATC sites in human or eukaryotic DNA. So this particular it is a hypothesis that the nicks which are present in the lagging strand synthesis, that is the Okazaki fragments which are not sealed as yet, these nicks are recognized by the MUT-S alpha and MUT-L alpha machinery and help in mismatch repair. And for the leading strand, the sliding clamp, which is known as pCNA in eukaryotes, sliding clamp is the protein which is recognized by MUT-S alpha and MUT-L alpha protein, and this helps in recognizing the strand which is having a mismatch, and it is later on processed and the mismatch is repaired. The other type of repair system is base excision repair system. Now, base excision repair system involves removal of a base from the DNA. Whenever a base is to be removed, the glycosidic bond which connects the base and the sugar has to be broken, and this is brought about by hydrolysis of glycosidic bond, which is carried out by an enzyme known as DNA glycosylase. Now, these glycosylases are very specific enzymes which recognize specific type of bases, modified or damaged bases, and they cleave only those particular damaged or modified bases. Some of the glycosylases can introduce a nick 3' to the site of the damage through their lyase activity. However, this particular activity is not essential for repair to take place. As can be seen in this particular example, a glycosylase cleaves the glycosidic bond which is connecting the deoxyribose sugar to its base, and this base is removed from the DNA. After the removal of DNA, there exists AP site. AP is apurinic or apyrimidinic site, and this particular site is also dangerous because it does not code for any information. So this particular site has to be mended so that it gets some information. Some DNA glycosylases have lyase activity, but it is not essential for the repair mechanism to take place. Now, after the excision of the base, AP site has generated. Now, there is requirement of endonuclease, which will make a cut 5' to the site of AP site. This endonuclease will cut 5' to the AP site. There are many types of AP endonucleases. In E. coli, 2 of the endonucleases are mainly in function. One of them has been named exonuclease 3 because initially it was categorized, its function was found to be that of exonuclease, but it had endonuclease function also. So, this exonuclease 3 is expressed constitutively in the E. coli cell, and the human function analog of this particular exonuclease is known as APE/HAP protein. This is expressed constitutively. However, there is another type of endonuclease which is known as endonuclease 4 in E. coli, which constitutes only 10% of the activity, but it is very much inducible by superoxide radical damage. Whenever DNA is damaged by superoxide radical, it leads to expression of this particular endonuclease. Now, both these endonucleases can make a cut 5' to the apurinic site generated by DNA glycosylase activity. After there is cut 5' to the AP site, there is another enzyme which is known as DRPase or deoxyribophosphodiesterase, which hydrolyzes this 5' residue, resulting in a single nucleotide gap. So, it is very essential to note that in base excision repair, there is only removal of a single nucleotide. After this nucleotide has removed, DNA polymerase can initiate repair synthesis. Sometimes it also happens that one cut is made 3' to the damaged site and a small chunk of nucleotide is removed, and then this is repaired. Whatever nick is remaining after the work of DNA polymerase, there is a nick remaining, it is again filled by DNA ligase. DNA ligase comes into play all the time because whenever a gap is getting filled, nick will always remain, and this nick will be joined together by DNA ligase. Now, this particular figure sums it up, the base excision repair machinery. There is a base which is shown in red, which is not an appropriate base, or it is a damaged base, and this is removed by DNA glycosylase. It is excised out, and there exists an AP site. Now, endonuclease which cuts 5' to the AP sites come into action, and it cleaves the bond. After that, DRPase hydrolyzes this particular nucleotide and removes it from its position. Now, DNA polymerase will come into action. They will introduce nucleotide complementary to the parental strand, and after that DNA ligase will seal the whatever nick is present there. What happens if the damaged base is not removed before replication? That is, DNA has got replicated and damaged base is still there. Sometimes it does happen. Then there are glycosylases known as fail-safe glycosylases in E. coli. These glycosylases recognize the aberrant base pairing. Suppose there is OXO-G which is base paired with A. It recognizes that there is some problem here. Though it won't be removing OXO-G, it will only remove A, and in place of A it will incorporate C in this particular site, so that there is no wrong information getting passed from one generation to another generation of the DNA. After that, it again gives chance to the normal glycosylases to act on the damaged bases to remove those particular damaged bases. In this case, OXO-G and A base pairing, A will get removed and OXO-G will be there. Normal glycosylase will then act on the OXO-G to remove it and make it a normal G, and this G will be pairing with C. Similar, another type of glycosylase exists which causes removal of T opposite a G. So, this T will be removed, in place of T, C will be incorporated. After that, this particular damage has been undone. Another type of repair mechanism is nucleotide excision repair mechanism, which is contrary to base repair mechanism, removes a large chunk of nucleotides from the DNA strand. Now, this requires presence of certain proteins which have been named UVR proteins in prokaryotes, and they have been named as XP proteins in eukaryotes. Now, the complex which functions here is known as UVR-ABCendonuclease because it requires presence of 3 protein ABC, or it is also known as excinuclease because it brings about excision of a stretch of nucleotide. Now herein UVRA protein is very important because it is the protein which recognizes the damage which has been done to the DNA. This particular protein also binds to ATP. After it recognizes the damage which has been done to DNA, it loads there and it helps in recruiting UVRB, which is again an endonuclease. After UVRB has been loaded to the site where there is damage, UVRA is removed from that particular site and it recruits another protein which is known as UVRC. Now UVRB makes cut 5 prime to the damaged site and it makes cut about 8 nucleotides from the damaged site, whereas UVRC makes a cut 3 prime to the damaged site. And how long cut it makes, that means whether it cuts 5 nucleotides away or 10 nucleotides away, that depends on the sequence. Usually it makes a cut about 4 or 5 nucleotides away, and therefore a stretch of nucleotide which is about 12 nucleotide long is removed. Here in UVRB is making cut 5 prime to the damaged side and UVRC is making cut 3 prime to the damaged side. UVRD is another protein which is a type of helicase which comes into action and it unwinds the DNA. After the DNA has been unwound and the damaged stretch of nucleotide has been removed, DNA pol 1 comes into action, it fills in the gap and ligase again seals the nick. Many a times it happens that this particular DNA is again getting transcribed, and whenever such DNA is getting transcribed, there is a transcription block here, and a transcription repair coupling factor comes into play here. It goes and binds to this particular damaged site and it recruits UVRA, UVRB, C to repair this particular damaged stretch of nucleotide, and that's why it is known as transcription dependent repair in E. coli. So in eukaryotes also there is nucleotide excision repair in function. There are similar proteins though they have different name, and these proteins are, as I already told, these are coming under XP category. There is a series of 8 to 9 proteins which function in nucleotide excision repair in eukaryote. Again there are proteins which recognize distortion in the helix, like XPC protein, which recognize that there is some distortion in the DNA and it loads there. After its loading, it recruits another protein known as TF2H. This TF2H is a very important protein because it's taking part in nucleotide excision repair and it also takes part in transcription. After XPC has been loaded and TF2H has been loaded, which and there is a loading of other protein known as XPB and XPD protein, which take part in unwinding of the DNA and the verification of the damage. Then other proteins come into play, and very important protein in this is ERCC1 XPF, which leads to incision at 2 sites, and this particular incision cuts out a stretch of nucleotide which has in between incorporated damaged nucleotide. After ERCC1 cuts the damaged stretch of nucleotides, then there are proteins which like DNA pol delta, which fill in the gap, and there is sliding clamp loader, and again there is filling of the gap and ligase seals up the protein. So there are homologous proteins in both prokaryotes and eukaryotes, though their names are different. Again there is transcription coupled repair mechanism also in eukaryotes as found in prokaryotes or E. coli. Herein, whenever a damaged strand is getting transcribed, there is a block of the RNA polymerase, it cannot move any further, and this is recognized by TF2H. TF2H then recruits all the proteins, XP proteins, which are required for damage correction, and this particular damage is corrected. So there is a global genomic repair mechanism which does not involve the RNA polymerase, and there is a normal mechanism which involves RNA polymerase blocking. These UVR proteins are needed to mend the damage from UV light in prokaryotes, and in humans they are needed for nucleotide excision repair of the thymine dimers. Whenever there are mutation in these XP proteins, it leads to type of cancer, cancer of the skin cells, and which is known as xeroderma pigmentosum. Whenever individuals which have mutant XP genes are exposed to UV light, then it leads to severe form of lesions or blotches on the skin of the individual, and this comes under a type of hereditary cancer. Another type of repair mechanism is recombination repair mechanism, which involves repair of single strand break or double strand break. The single strand break repair requires filling of the gap with the homologous region of sister duplex, and it is also known as daughter strand gap repair or post replicative repair. Double strand break repair requires sealing of the two strands of the DNA which have broken, and it could be crossover repair where because of recombination crossover products are formed, or it could be non-crossover repair mechanism, which is also known as synthesis dependent strand annealing repair, and it is usually seen in p element transposition. The daughter strand gap repair requires Rec-A protein. Rec-A is a protein which is involved extensively in repair of the DNA, also in recombination of DNA. So, Rec-A protein is loaded onto the gap created on the newly synthesized strand. Whenever there is a lesion in the parental strand, there will be a gap in the daughter strand which is getting synthesized. So, in this particular gap, Rec-A protein will be loaded by Rec-4 protein. After loading of Rec-A and Rec-A by Rec-4, there will be invasion of the parental strand of the homologous chromosome, and this invasion will be followed by branch migration, which will be held by RUV, AB and Rec-G protein. After this branch migration, there will be gap filling DNA synthesis, and then the holiday junctions will be resolved. There are two holiday junctions which can be seen here as crosses in the right figure at the topmost position, and after these holiday junctions have been resolved, there will be gap filling DNA synthesis. So, we are getting a normal DNA, and there can be crossover also, so that the lesion gets transferred to the sister duplex. But where there was gap previously, now there will not be any gap present here. This is known as daughter strand gap repair. Another mechanism is double strand break repair, as can be seen in the figure that both the strands of DNA they are broken. So, it requires that both the strand anneal to the specific position. Herein, certain proteins known as mRNA protein and ATM protein are required. mRNA protein is a protein which screens or senses the double stranded break, and it leads to activation of another protein known as ATM protein. After these proteins are loaded, there is resection of the DNA strand. Resection is removal of nucleotides from one end. After the resection, there is creation of single stranded DNA tails, as can be seen in the figure, and these tails are loaded RAD51 protein loads onto these proteins. After RAD51 has loaded, with the help of the homologous chromosome, there is invasion of one of the strand of the DNA, or there could be invasion of both the strands, and there will be complementary nucleotide addition and complementary DNA strand synthesis. There are two holiday junctions here. There can be formation of crossover after resolution of the holiday junction, or there can be normal products also. All in all, after the double strand break repair has functioned, it leads to formation of the normal DNA which does not have any break anymore. So, this is a very important mechanism of repairing. Now, another mechanism is synthesis dependent strand annealing. Synthesis dependent strand annealing does not produce crossover products. It produces normal products, whereas double strand break repair might produce crossover products. In synthesis dependent strand annealing, again there is a double stranded break in the DNA, as can be seen in the green lines. These are broken DNA molecules. There is invasion, first strand invasion into the homologous sister chromosome, and this leads to formation of the complementary DNA strand. There could be second strand invasion also, leading to formation of a second complementary strand. after a stretch of nucleotide has got synthesized, there is complementary base pairing between these two newly formed strands, as can be seen in the figure. The red strands are complementary base paired, and then after complementary base pairing, there is extension of the DNA by DNA polymerase. Gaps are filled, and again there is sealing of the gap by ligase. So this particular mechanism of repair does not lead to crossover product getting formed. It only leads to normal product getting formed. Sometimes it happens that the damage to DNA is extensive, and normal DNA repair mechanisms like base repair mechanism, nucleotide excision repair mechanism, or double strand break repair mechanism, they cannot function properly. So for that particular condition, there is another repair mechanism which come into play, that is known as non-homologous end joining mechanism, or NHEJ mechanism. This particular mechanism uses certain proteins, of which Ku70 and Ku80 are protein are very important. Ku70 and Ku80, which are represented here by green fluorescent green protein, they recognize double stranded break in the DNA, and they load onto DNA, leading to recruitment of another protein, DNA PK, which is a kinase. After DNA PK has loaded onto this particular region, there is loading of Artemis protein, and this particular Artemis is activated by the kinase activity of DNA PK. Now this Artemis leads to formation of the resection of the DNA ends, and after that DNA polymerase is loaded and the gaps are filled, which leads to formation of nick, which is again joined by ligase. Now this particular non-homologous end joining mechanism might lead to a mutation, serious mutation. But this mechanism is better than not having a normal DNA at all, because if any organism is having a broken DNA, then if there is replication or transcription, then all the information has been lost and the machinery will get stalled at the broken end. But non-homologous end joining ensures that the two ends are broken. There might be information which has got lost during this particular repair mechanism, but still the DNA polymerase which causes replication of DNA, or RNA polymerase which causes transcription, it is not stalled, and it will move on further from the point of break, so that other genes which are present downstream, they can get replicated or they can get transcribed. It has been found in eukaryotes that this particular non-homologous end joining mechanism also is involved in formation of various types of antibody during VJ recombination. This particular mechanism occurs less frequently in prokaryotes, and it occurs more frequently in eukaryotes. However, it is found in some organisms like Bacillus subtilis. Bacillus subtilis produces a Ku-like protein and a DNA ligase, and it packages them along with the DNA in spores. Any spore which is a mutant for this particular Ku protein will be susceptible to dry heat, because its DNA will get damaged, but it will not get repaired. Whereas a normal Bacillus subtilis spore, which has a normal Ku protein and DNA ligase, whenever it is subjected to heat, even after DNA damage, even after there is double stranded break in DNA, this Ku protein and ligase protein will ensure that the double stranded DNA gets rejoined together, and it can germinate afterward. The Bacillus subtilis uses this particular mechanism because it does not have any homologous chromosome. It has only one chromosome, so it cannot rely on double strand repair mechanism or other mechanism which require recombinational repair. Another type of repair mechanism is translesion DNA repair, which comes into play when there is severe damage to the DNA. This translesion DNA mechanism requires presence of translesion polymerase, so that this translesion polymerase, whenever it encounters a replication block, it ensures that certain nucleotides are filled in the complementary strand. They might not be normal base according to the normal base pairing. For example, whenever there is thymine dimer, it ensures that certain bases are incorporated, whether they are G, C or A, so that there is continuity in the complementary strand and there is no more replication block or transcription block. It has been found in E. coli that there is translation DNA synthesis it is done with the help of OMUC polymerase, which is a translation DNA polymerase of Y family. This particular type of polymerase is found in all the domains, but it carries DNA replication with low fidelity, but it has the ability to bypass lesions which are present in DNA. So OMO-D and OMO-C complex helps in incorporation of bases complementary to a DNA lesion, and this requires presence of another protein known as DIN-B. Eukaryotes also have similar proteins, homologous proteins, which also function in a similar way, and this helps in overcoming whenever there is severe damage to DNA. All the eukaryotes have translesion DNA synthesis mechanism. Now, how does translesion polymerase gain entry into the stalled replication machinery? It is a very important issue, and this translesion DNA polymerase gains access with the help of sliding clamp protein. Whenever sliding clamp protein gets ubiquitinized, that is a molecule of ubiquitin protein is added to sliding clamp protein, then translesion DNA polymerase recognizes this particular sliding clamp, and it gets incorporated there, and it removes the normal replication machinery, and it starts translesion DNA synthesis. After the gap has been filled, after there is incorporation of normal nucleotides, it moves away and the normal replication machinery come into play. This translation mechanism comes into play during SOS response, that is when there is severe damage to the DNA. When there is SOS response, Rec-A protein gets activated. Rec-A protein gets activated whenever there is single stranded DNA region in the organism. There is another protein known as Lex-A. Lex-A protein is bound to the proteins to the regulator regions of the genes which take part in SOS response. Whenever there is severe damage, Rec-A gets activated. This Rec-A leads to cleavage of the Lex-A or degradation of the Lex-A protein, so that the protein which were earlier genes which were earlier repressed are now activated, and SOS response genes get activated, so that the repair mechanism comes into play. However, during activation SOS response, activation of Rec-A can also induce activation of certain repressor of bacteriophages, and this causes removal of bacteriophages from the lysogenic state, and they become active and they start their lytic cycle, which can lead to death of the organism or E. coli. There are many diseases which are associated with mutations in DNA, DNA repair machinery, and some of these, as I have already told, are HNPC, which is occurring because of damage to the mismatch repair system; Xeroderma pigmentosum, which occurs whenever there is defect in nucleotide excision repair mechanism; Bloom syndrome, which occurs whenever there is DNA ligase deficiency; and diseases like Huntington's disease and Kennedy disease, which occurs whenever there is slippage in the DNA replication machinery. Now, how do we detect mutations? Mutations can be detected in bacteria and fungi through the use of technique known as detection of nutritional mutants. Many fungi and bacteria have a mutation in genes which code for essential enzymes or essential amino acids. These can be detected by various culture techniques, and Beadle and Tautom use this particular detection technique to frame their one gene one enzyme hypothesis. Mutations can be detected in Drospohila also, and Mueller detected mutation in Drospohila using CLB method, which is a very common method taught in genetics class, wherein X-ray were used to induce mutation in the males of a generation, and after that progenies were followed, and it was seen whether X-ray induced any mutation or not in this particular X chromosome of the male. For this particular discovery, Mueller was awarded Nobel Prize in Physiology in the year 1946. In humans, mutation can be detected using pedigree chart. It's a very well-known chart of the hemophilia disease in Queen Victoria family, and pedigree charts can be used to find out in which particular generation mutation has occurred, and how it has passed on from one generation to another generation. Nowadays, cell culture techniques and molecular techniques can also be used to find out which gene codes for which protein and how mutations occur, in under various circumstances. The mutagenicity of chemical can also be assessed by using a test known as Ames Test. Ames Test helps in finding whether a chemical is mutagenic or not, and when a chemical is found to be mutagenic, it can be assessed for its carcinogenicity. Ames test uses a strain of bacteria known as Salmonella typhimurium, which requires histidine for growth. Whatever chemical we are testing for mutagenicity, we incubate that with the Salmonella culture, which is growing in a medium which has minimal histidine. If there is any mutation induced, then there will be reverting back of this particular Salmonella strain to normal strain, and it will start growing in this particular minimal medium. Whereas if there is no mutation, then all the cells will die. Greater the mutagenicity of the given chemical, greater will be the number of colonies formed after mutation. For testing any chemical for carcinogenicity or mutagenicity, it is incubated with rat liver extract, because in eukaryotes it happens that in the higher organisms, liver enzymes lead to detoxification, and in this process many chemicals which were earlier harmful turn mutagenic. So this particular test can find out whether chemical is mutagenic or not. Mutation has been used extensively in research also. There is site-directed mutagenesis, which can induce desired changes in the gene, and effect can be found out. And there are knockout mutation where a gene is turned into a non-functional gene, and then its phenotypic effect can be found out. So this figure shows that there can be introduction of specific changes in the DNA, and it can be found out what these changes lead to in the change of in the structure and function of protein. Also, knockout mutation lead to change in the lead to induction of a non-functional or null allele in the organism, and changes can be found out. Many diseases have been worked out by using this particular technique. This is the figure which shows how the knockout mice are generated. Michael Smith was given Nobel Prize in Chemistry for site-directed mutagenesis in 1993, and 3 scientists were given Nobel Prize in 2007 for creation of knockout mice. In this particular year, that is 2015, the Nobel Prize has been given to 3 scientists, namely Thomas Lindell, who found out about base excision repair, Aziz Sankar, who found out about nucleotide excision repair, and Paul Modric, who found out about mismatch repair. After these announcements, new class of DNA repair enzyme has been found out, which is a glycosylase, and which does not require flipping of the base of the damaged base. So I hope that more DNA repair enzyme continue to be found out, and more mechanisms continue to be found out in various kinds of organisms, so that we can find out the causes of different mutations leading to formation of cancers, and it will bring about some amelioration for the human race as such. So thank you very much for the lecture.